that 2 of your lecture exams would be worth 110 points and 2 would be worth 120. This is the second one that's worth 120. And it's because we have a lot of important stuff in Unit 4, so we devote a little more testing to it. There will be a written section at the end as before, but we're going to start with multiple choice true false Scantron based section, and that'll be 80 points, and then the written part will be the remaining 40 points. After the test, we're going to do our lab review, but that's going to be pretty brief, so I'm going to show you the last extra credit video. And so after you take your exam, you step out until noon, come back at noon, we'll have the lab review, and then we'll have the extra credit opportunity. And that's only for people who stay here. There's no opportunity to take it home and work on it later. Just because too many people, one person watches it and gives the answer to 5 other people and they don't all see it, and that's not my goal. My goal is you see the videos. So extra credit opportunity after the exam, so don't make plans. If you want those 10 points, don't make plans for leaving early on Tuesday of next week. Okay, so the beginning questions are all about phylum Chordata, and we have that chart, and so remember, we talked about evolutionary advances, and they're going to feature in this test. We talked about the evolutionary advances of the chondrichthyes, the cartilaginous fish, versus the previous group, that they had paired fins and a jaw, and that allowed them to swim better and bite things so they could get more energy-rich food, eat other fish. More energy allowed them to compete better. We had evolutionary advances like the swim bladder and the operculum and the bony fish, the lobe fins and the lobe fin bony fish. We had the legs in the amphibians. And then for the reptiles, our advantages included the main evolutionary advance was the amniotic egg. And that was the first group to have the amniotic egg, which is an egg that can be laid on land because it has a chorion, which allows gas exchange without very much water loss. And that was what allowed the vertebrates to really colonize the land successfully. And only those 3 reptiles, birds, and mammals, the amniotes, the ones with the amniotic egg, those are the only ones that are fully successful on land and can live in desert environments. Remember that in our subphylum Vertebrata, we have all the chordate characteristics: postanal tail, notochord, gill slits, and dorsal hollow nerve cord. That's in our phylum Chordata, but vertebrates typically have vertebrae, which is a backbone, but one class didn't. So we can't even say that all vertebrates have a backbone because the Mixini lost theirs, the hagfish. But remember those characteristics and their basic meaning. We've got the jaw and the paired fins, and I've already talked about that. I'm just looking at my notes here. You should remember cartilaginous fish and like their skeleton cartilage. What is cartilage? It's chondrin and collagen, and they are, but they don't have things like swim bladders and an operculum. We talked about the lobe fin bony fish, class Sarcopterygii, as being slightly adapted to life on land because 5 of the 8 species have to breathe air. If you have to breathe air, that's an adaptation to land life. And they can stump around on land, and their skin's waterproof enough that they don't dry out very fast, and they can spend a fair amount of time on the land, even though they're considered aquatic, not terrestrial. Remember that that group, Sarcopterygii, is slightly adapted to life on land. Whereas amphibians are more adapted, they're called partially adapted, and then the reptiles, birds, and mammals are considered fully adapted to life on, completely adapted to life on land. I'm remembering the chart as I'm thinking here. So again, the chart features quite importantly on this exam. Remember, amphibians use their body surface or skin, their gills, or their lungs at some point in their life, all of those gas exchange surfaces. Remember the ray fin bony fish or Actinopterygii, the lobe fin bony fish or Sarcopterygii. Okay. Remember that only two groups are endothermic, and that is birds and mammals. And even though there are some exceptions, the vast majority of all the other groups are ectothermic. And remember, ectothermic means your temperature is set by the environment around you. It's what everyday people call cold-blooded. I've said why that term isn't really accurate. But if you just memorize birds, mammals, endotherms, warm-blooded if you prefer, and then everything else is ectothermic, and that'll help you because several times I ask you, is this group ectothermic or endothermic? It's on your chart. So if you just memorize birds, mammals, endotherms, everything else ectotherms, you won't go wrong. Okay. Let's see. Remember that all the chordates are deuterostomic development, not protostomes. The vertebrates that are alive today, sort of the most primitive version, the earliest version that still has some descendants surviving today, are the jawless fish. So those would be classes Myxini and Petromyzontida. They have the least of these evolutionary advances. They don't have the paired fins or the jaw or any of the other things I've been mentioning. Okay, remember the Actinopterygii and everything after them has bone skeleton. The bone is made of calcium phosphate and collagen. And that the lobe-fin fish evolved into the amphibians, and the amphibians evolved into the reptiles, and the reptiles evolved into mammals and birds eventually. Remember which group has a, which two groups have a four-chambered heart. That would be birds and mammals. Remember though that mammals typically, most mammals give live birth, don't have an amniotic egg, and have a placenta. But birds don't have a placenta, but they do have a four-chambered heart. It may seem like a weird set of things, but it's because my notes indicate I should say that. Okay, and when you see the test, you'll probably get what I'm talking about. Then we have, I've already mentioned the first group to not have to lay eggs in water was the reptiles, Reptilia. Remember the origin of the swim bladder was an early lung that helped creatures in low oxygen water get enough oxygen, and it began to be used as a buoyancy organ, so they could float at any level without moving. And so remember that origin of the swim bladder. It was originally a primitive lung. And some fish kept it as a lung and use it to breathe air still, like the lobefin fish. The only terrestrial group that doesn't have the amniotic egg are amphibians. Amphibians, even though they require water to reproduce, they still are considered terrestrial or land dwellers. But the other three groups of chordates are all terrestrial and do have the amniotic egg, or like us, something that evolved from the amniotic egg, our extraembryonic membranes in the womb. Remember that of these subphyla, we had Cephalochordata or Urochordata, and I said one of those is more likely the ancestor of vertebrates than the other. So look that up. That's at the beginning of the notes on phylum Chordata. Okay. Remember, all chordates have a dorsal hollow nerve cord, the other three characteristics, but not all of them have a backbone, so they're not all vertebrates. There are some invertebrate chordates, Urochordata and Cephalochordata, for example. Remember, the lateral line system is found in the fish. We introduced it in cartilaginous fish, and it's shown in the image of a bony fish at some point in your materials. Other fish I think other fish have it too, but certainly it's in the cartilaginous fish and the bony fish. So remember, and it's a pressure sensing structure that helps them get information about their surroundings when the water is too clouded with sediment for them to see what's around them. Okay, we talked about the operculum helping to ventilate the gills without moving, how that helps an animal survive. Remember that reptiles, birds, and mammals have this better breathing. It's called negative pressure breathing, where the chest cavity creates a negative pressure and sucks air in. but amphibians have positive pressure breathing where they put the air in their mouth and push it down into their lungs, and that's less efficient. Remember that positive pressure goes with amphibians, negative pressure goes with the other three land-dwelling chordate groups. Okay, we talked about the chorion already. Remember, we already talked about which fish have jaws and which ones don't. Remember the role of keratin. It helps make the skin of reptiles, birds, and mammals waterproof. It also is involved in making feathers and fur and fingernails and other things that we've talked about. So keratin is one of those molecules that is important in chordates, and I want you to kind of pay attention to where it occurs. And I've got it in the notes, wherever it's highlighted. I intend to ask a few questions about it. Okay, so talk a little bit about the urinary system for a while. Remember that nitrogenous wastes come from the breakdown of amino acids for cellular respiration, and that animals can excrete either ammonia, urea, or uric acid as nitrogenous wastes. Remember that Remember that uric acid costs the most energy and the least water. Ammonia costs the most water and the least energy, and that urea is in between the two. Remember that when water and small molecules are pushed across the walls of the glomerulus, that this is called filtration. And then the filtered fluid goes, which is the forming urine, goes into the Bowman's capsule to begin its journey through the nephron. Also remember that filtration is powered by blood pressure. That's the pressure that pushes the water and solutes, small solutes, across the filtration membrane of Bowman's capsule. When you put stuff from the urine back into the blood, that's going to be reabsorption. You can also put some bad solutes from the blood back into the urine without doing filtration, and that's called tubular secretion. But the major, that is somewhat important, but most important is filtration and reabsorption, leaving the bad stuff behind. And then secretion allows you to add a little bit more bad stuff to the urine. And as I mentioned in that lecture, remember that our urine can be hypotonic, isotonic, or hypertonic to help us deal with osmosis issues and things like that, and dehydration and such on land. Hypertonic urine helps you save water, is the bottom line. You should remember about the kangaroo rat that it has 4 ways of holding on to water better than we do, like waterproof skin and fur, dry feces, dry exhaled air, and the most hypertonic urine in nature, with least water. And it gets all its water from cellular respiration. This water is called metabolic water because it's made by metabolism. So I'm going to ask you where it gets its water from, cellular respiration or metabolic water. And then I'm going to ask you, I'll probably list those 4 ways it holds on to water better than we do, maybe list 3 correctly and 1 incorrectly and have you pick the wrong one. Or I'll list 1 correctly and 3 wrong ones and have you pick the right one. But don't forget the K-rat story. Moving on to the nervous system, I had the reflex arc for you. The arrow A was the stimulus. Then we had a sensory afferent neuron taking the information to the interneuron, where we have information processing going on, and the interneuron is a neuron in the central nervous system. And if it decides to respond, it's going to send the signal down the efferent neuron, the effector neuron. If it's going to a muscle, that effector neuron is called a motor neuron. And that was D in your diagram. And then E was the muscle. So this is the reflex arc that I had. And as I said, there'd be a number of questions. There looks like about 6 questions on that, what the things I just went over verbally here. Okay, remember the resting potential, that the resting membrane is at this resting potential because of the pump and the potassium leak channel. And so the membrane is leaking potassium out to make the inside negative membrane potential. And the resting potential is more negative, and when you raise it up to threshold, make it more positive, then you can get an action potential. And at this resting state, the concentration of sodium is high outside and low inside, and the concentration of potassium is high inside and low outside. If you know the gradients, it makes everything easier to understand. I haven't urged you to memorize the gradients. I mentioned it, but if you memorize them, it makes it easier to understand everything else in the lecture. Okay, remember, again, if an excitable cell like a neuron or a muscle fiber is not carrying signals, that's at its resting potential. And that a threshold potential is the minimum depolarization necessary to cause an action potential. And the action potential begins in the part of the neuron known as the axon hillock. Now, in the last lecture, Tuesday's lecture, I had the action potential XY plot on the screen. I had the number 1 next to the y-axis, which is membrane potential measured in millivolts. And I said I'd ask you, what is it and what are its units? Number 2 was by the x-axis, which is time measured in milliseconds. And I'm going to ask you, what does the x-axis measure and what are the units, time, milliseconds? So that's 4 points right there. Number 3 was the threshold potential. Number 4 was the resting potential. Number 5 was our cell being stimulated by another cell. Number 6 was when our cell reached threshold and the voltage-gated sodium channels opened. Number 7 at the peak was the voltage-gated sodium channels closing, the voltage-gated potassium channels opening, and then number 8 along the far side was potassium rushing out of the cell. That right there is it's the exact same test question, same picture I had on the board and everything. Numbers are all in the same place. Okay, and that is going to be 11 questions on the action potential diagram. Okay, remember for muscle that when a muscle contracts and the load moves, that means the length is shortening and tone is not changing, so that's isotonic. When a muscle contracts and the load doesn't move, then length is staying the same. It's not shortening because the load's too heavy to lift, but tone is increasing. So that would be isometric. At least one of those will be on the test. Remember what a sarcomere is. It's a unit of muscle contraction in the myofibril. I already mentioned the neurotransmitter used at the neuromuscular junction is always going to be acetylcholine. Remember that neurotransmitter for both the synapse in Tuesday's lecture and today's neuromuscular synapse, the neurotransmitter is stored in the presynaptic cell, and an action potential there causes the exocytosis of neurotransmitter. And I don't think it's on the test, but it might be that exocytosis of neurotransmitter is caused by a rise in calcium levels. Calcium signals that. Just like calcium allows muscle contraction, calcium is a signal. Let's see. And then the neurotransmitters are released by exocytosis, and then the receptors for the neurotransmitters are on the postsynaptic cell, which is the muscle fiber. Okay, remember that a sustained contraction by rapidly repeating action potentials is tetanus, and a single brief contraction caused by a single muscle action potential is called a twitch. Remember that muscles relax. I said that the head detaches, and the head detaches when ATP binds. So we'll go back up here. So ATP binds, then the head detaches, it's released. And here it's attached, there it's not. So if you didn't have ATP present, like a person, a mammal, well, a chordate, muscle all works pretty much the same in all the chordates. If the chordate were to die while the muscle's contracted and stop making ATP, then there wouldn't be any ATP to bind to get the head to release. So a lack of ATP can prevent the muscle from relaxing. The muscle remains rigid. Okay, now don't forget where calcium binds in the story. It binds to troponin. You don't need to know troponin C or any of the other troponins, but just troponin. And then the thing that blocks the myosin heads from binding is tropomyosin. And that's what troponin moves out of the way when calcium binds to it. Remember, the impulse that triggers the contraction is an action potential that travels deep inside through the T tubules. For our sarcomere diagram, I said earlier today, but now it may make more sense, which letter shows the place that's made of myosin? A. Which letter shows the place where ATP binds? A. Which letter shows a place where there's a a binding site for actin. Well, that's on the myosin head. And then I could say which letter shows the place where calcium binds, and that would be C, the troponin of the thin filament. Which one is made of actin, and that would be the thin filament as well. Where are troponin and tropomyosin located? Also C. So made of actin is C as well. I misspoke there, but anyhow, all those things are possible. The Z line is B. The whole sarcomere is represented by the letter D. As I mentioned, the letter E is where the two overlap, so you can get this cross-bridge formation. And that pretty much covers that. For the cross-bridge cycle, which I went over on the board here, I mentioned arrow D is where ATP binds, arrow A is where ATP hydrolysis occurs. Arrow C is power stroke, it's also where ADP and phosphate are released. So those are, the picture's gonna look exactly the same as the one in your notes. Now on to the written section. Again, this will be like the written section in exam 3, where in that exam, the written section had a number 1, and then it had a question to try and get you to name a phylum and spell the phylum correctly. Well, this time I'm going to have a leading question at the beginning of each section that's going to have you identify a class. So I might say, this class developed the operculum and the swim bladder, and you'd put Actinopterygi in there. And then the remaining questions would be about that class. So what kind of questions might be here? Well, some of these, certainly for any fish group, I might ask, is it a single loop or double loop circulation? And whether it's a 2, 3, or 4 chambered heart. For birds and mammals, 4 chambered, fish 2, reptiles and most reptiles and amphibians, 3 chambered heart. You can remember too that urochordates, now this is not in this section but elsewhere, I kind of missed it, but urochordata, the tunicates, undergo complete metamorphosis, and so do amphibians. Amphibians are land dwellers. Some students don't really feel that way because of their necessity to get water for reproduction, but they definitely live on land. They're the first group to spend most of their lives on land. And if I ask about the excretory organ for any chordate, there's only one answer: kidneys. You should know all the respiratory surfaces from the chart for each group. So if it was amphibians, it'd be skin, body surface, gills or lungs. For birds, mammals, lungs only, so on. Remember that amphibians have positive pressure breathing. I already said that, but the three amniotes have negative pressure breathing, which is better. And remember, amphibians, of course, are not fully adapted to life on land because they have to lay their eggs in water. And they also need, they can't have a waterproof skin, most of them, because the gas exchange through their skin. Some have evolved a somewhat waterproof skin and a better ability to live away from water, but they still have to lay their eggs in water. Remember the role of keratin in the waterproof skin. Remember, in the egg, you have not only the chorion, which allows gas exchange without water loss, but also the yolk sac, which stores food. Yolk, food, remember that. Allantois, or allantois, I don't know, tois, I think is how the doctors say it. That is going to store waste. I won't ask about the amnion, shock-absorbing fluid, but the chorion, the yolk sac, and the allantois, you should be comfortable with what they do. Remember the common names for the classes, if there is one. So that would be mainly aves is birds, Actinopterygi is ray-finned bony fish, but if you just write ray-finned fish, that's fine. And for Sarcopterygi, it's lobe-finned bony fish, but lobe-finned fish will be fine. Remember, if I ever ask about their body temperature, if it's not a bird or a mammal, it's going to be ectothermic. And remember the evolutionary adaptation. So if I'm asking about the bony fish, for example, it's the ray-finned bony fish. It's going to be the swim bladder and the operculum, for example. For the reptiles, amniotic egg, among other things. Remember that both birds and mammals have the best hearts of any chordate or any vertebrates, but that bird lungs are actually a little better than mammal lungs. Who has the best lungs and heart? Well, birds and mammals are tied for the best heart, but if you add lungs in, birds have the best because they have the highest metabolism. They need the most oxygen per minute to survive. Now, if I ask about birds and mammals and a structure made of keratin that helps keep in body heat, it's going to be feathers for birds and hair for mammals. If I ask for a unique feature that helps these creatures maintain body heat, it's got to be feathers for birds and hair or fur for mammals. It can't be fat under the skin because that's not unique to one group. Both birds and mammals have fat under the skin. So I ask for a unique feature that helps them keep in body heat. Feathers for birds, fur for mammals. And the others don't need to do that because they're not endotherms. Remember the adaptations that birds have for flight, including no teeth, hollow bones, feathers, and feathers are really an easy one to remember. Remember the mammals. The mammals include monotremes, those are the egg layers like the platypus, marsupials, the kangaroos and possums, and placentals, which are most mammals, including humans. And they use hair to keep in their body heat. If I ask you what they give their young for food, please don't say mammary glands, say milk, because that's actually the food. But many students seem to answer that question with mammary glands. I'm not sure why. Remember that both birds and mammals have a lot of parental care. Unlike the other groups that lay huge amounts of eggs and ignore them, birds and mammals have fewer offspring and care for them. So higher degree of parental care. Either way, you're going to get some surviving. Either a few out of many with no help, or just make a few and help them. Either way, enough will survive to keep the species going. Also remember the placenta. That's where exchange of materials happens between the fetus and the mother in most mammals, in the placental mammals. A lot of students want to put umbilical cord for this, but that just takes the blood to and from the placenta. The actual exchange of materials happens in the placenta. Remember for the Sarcopterygi that there are two orders, Actinistia, which you don't need to know for the test, and Dipnoi, which you do because this includes members, the lungfish, that must breathe air. They have to breathe air. They're called obligate air breathers. They're obligated, or they have to breathe air. Remember, well, that's probably enough for that. Finally, something you're probably not prepared for, but it's in the Canvas notes and it's easy to look up, and the Canvas notes for Unit 1 are still open. And I may have mentioned this in Unit 1. When I talked about Darwin's theory of evolution, I gave you 5 summary statements. I asked you to list them for me, like more offspring are born into a generation than will survive and reproduce. Or number 2, there's variation among offspring. And number 3, offspring with certain characteristics have a better chance of surviving and reproducing than offspring with other characteristics. And then number 4, some of these characteristics that result in differential survival and reproduction are heritable. And number 5, vast bands of time have been available for change. I'm going to ask you to list those again. If you want, I can run up, while you're watching the dissection video and preparing for dissection, I can run up a hard copy for you. Just ask me. Otherwise, look in your Canvas or your Unit 1 notes if you still have those. But my goal here is to get you to think about evolution one more time before you leave the class. Maybe cementing those ideas so that you have better access to them in the future, because I, as a biologist, for most of my life, ever since I was about 15 years old, I've loved biology, and I still get new insights about evolution almost every day. It's such a powerful idea that I want you guys to think about it one more time. So I'm going to have you write down the 5 summary statements. And that's it for this exam. You can come grab your phones.